So perhaps we'll start with the definition of what a neoantigen is. And it's an antigen that arises as a result of somatic mutations that are acquired by cancer cells. These mutations are then translated into mutant peptides and have an opportunity to associate with MHC class 1 molecules, and for this peptide MHC complex to be presented at the cell surface to T cell receptors. And there is an opportunity then, if there is a T cell receptor activation, for immune response to be generated against cells that bear that and present that particular neoantigen. And the reason why the neoantigen hypothesis is so powerful in the context of melanoma is because melanoma is a highly mutated type of cancer. And of course, this is to do with a fundamental role of UV light in driving the risk of melanoma. UV is highly mutagenic, so melanoma cells will have accumulated up to 100,000 genome-wide mutations as a result of mutagenesis that's caused by UV. And the opportunity to form a neoantigen is really a probabilistic process. So the more mutations there are, the more likely it is that that one mutation will just have the right characteristics. One, to bind the MHC class 1 molecule with a higher affinity than the native peptide would, and two, to encounter a T cell receptor and escape and lead to an immune response by means of escaping central tolerance. And what's really relevant here is that this mutant peptide needs to be fairly distinct from the wild type peptide. If it's too similar, it will simply not lead to an immune response because of the mechanisms of immune tolerance. So the more mutations there are, the greater the probability that one of them will result in a neoantigen. And what we know about the neoantigen landscape so far in melanoma is that it does associate fairly well with the response to Immuno-Oncology agents, and namely immune checkpoint blockade. So we know that the higher that tumor mutational burden, the more likely it is that the patients will benefit from immune checkpoint blockade. So what that is telling us is that there is an endogenous priming of T cells by these neoantigens, which leads to upregulation of immune checkpoint proteins, and therefore the addition of immune checkpoint blockades then releases the possibility of those T cells to result in effective anti-tumor response. And then, so that's more from the biomarker point of view. And of course, what the subject of this session at the Congress is actually exploitation of neoantigen concept for therapeutic targeting. And there are really three different ways in which that can be achieved. One is to mimic these neoantigens in the form of vaccines. The second is to extract patients' tumor-infiltrating lymphocytes, so the T cells that are presumably responding to some of these neoantigens, and enrich them for those T cells that are responding to the most potent neoantigens, expand them, infuse them back into the patient. And lastly, it's through an engineering of those T cell receptors that are likely to recognize a specific neoantigen. So the neoantigens have really lent themselves to a broad number of therapeutic approaches, all of which are now in development.